Fighting Blindness Canada's Viewpoint is an education series that brings you the latest in vision research presented by health experts from across Canada. The session you're about to watch is a recording. To learn more about the research we fund and upcoming programs and events, please visit our website at fightingblindness.ca. Viewpoint is proudly presented by Bayer and supported by AbbVie, Apellis, Astellas, Beacon Therapeutics, Biogen, Johnson & Johnson, Novartis, and Roche. Thank you for watching. We hope you enjoy this session and share your thoughts with us in the comment section below.
Well, it's great to be with you today. Thank you very much for the very kind invitation and all the work that was needed to put this together. I'm going to stand up here so I can actually see what I'm talking about here on these slides, and I will definitely take you through those. So thank you for the very kind introduction. So I'm a Korean-American male, about 61 years old, about 5'6". I have probably a few more pounds than I should. I'm in a dark blue suit and very graying hair. So that gives you an image of who is speaking here. But I just want to talk to you a little bit about something called gene-independent or gene-agnostic therapies, which is relatively a new realm that has really picked up steam lately. And there are a number of ways that we actually do this. By way, I do just because I am an employee of an industry partner or an industry group, I just need to have some disclosures. I am an employee. I do some consulting for Spark Therapeutics, and I'm on a number of scientific advisory boards. So in the IRD space, right here there's a graph that basically shows that in recent years, there's really been an explosion of how many genes have been really identified and cloned, upwards of 300. And so it's really a rich area for research and for investigation into genetic therapies. But the idea is that right now, we have one therapy. So on this slide, there are a couple videos here where it shows a dog that has the shaking of the eyes known as nystagmus, and one of our patients that was in the clinical trial that has the same issue with the eyes moving back and forth. And what this is, in clinical research, we tried to use animal models to make sure the product that we're working with is safe and that we have enough evidence to go to regulatory bodies that they'll give us a go-ahead to then go into human trials. So RP65 disease is a relatively rare form of what we call early childhood retinitis pigmentosa or Leber congenital amaurosis. And because there are so many different names to this diagnosis or so many diagnoses to this disease, we call it RP65-related inherited retinal disease. And this whole development really started in the late 1990s when when the gene was discovered, and then through preclinical trials with animal models, it went on to human trials, and then in 2017, it was approved by the US FDA. In 2018, it was approved in the European Medicines Agency. 2020, it was approved by Health Canada, I believe, and then the first patient was injected, I believe, in 2023. Luxturna is what the trade name is, and it's currently approved in over 40 countries around the world. And you may still see some articles once in a while concerning its use. So that's what we have right now in gene therapy, and this is what we call a gene-specific therapy. In other words, you have to have a misspelling or what we call either a variant or mutation in the RPE65 gene to qualify for this treatment. And unfortunately, it's great if you have that gene because you can have this gene therapy, but it really only accounts for about 1% of adult RP and only about 10% of Leber's congenital amaurosis. And you'll hear different numbers, anywhere between 6 to 18%, but that's kind of what we use. So that means there's over 300 other genes that still do not have any type of treatment. The reality is we don't have the resources, the manpower, or the dollars to go to every one of those genes. And this is where gene-independent programs may come in. And there are a couple different kinds of things that you may hear. Now, just to finish on Luxturna, one of the things that we did was we used this maze. And basically what is on the screen is there's this maze, and what is on the maze is there's this arrows on the floor with obstacles on the side, and our subjects had to go through this maze at different light levels. And the whole idea was, can we make the retina a little more light sensitive that they can do more activities of daily living and therefore have a better quality of life? And what we were able to demonstrate was that before treatment, what this subject is right now, he's going all off the course. He's walking on the obstacles, and we have to put him back on and things like that. But a year after, we do the same patient, slightly different course, and this time he's walking right where the arrows are, he's not hitting any obstacles, and he basically has a perfect score. And this is basically the way that Luxturna was approved. This is the primary endpoint. And so that's kind of the way the story ended, and now it's as I mentioned, in many different countries. So when it comes to gene-independent or gene-agnostic therapies, there are a couple different ways that we do this. There's something called optogenetics, there's something called modifier genes, and then there are neurotrophic factors. I'm not going to go into each one in detail today because I don't want to put everybody to sleep, but it's one of those things that we are going to try and give you a little snippet so you can at least start the conversation. So optogenetics is pretty much in the headlines, many ways. The number of groups are going after this. And optogenetics is when in the retina there are cells that sense light naturally. That's their main job. But in inherited retinal disease, that's one of the primary cell types that goes through degradation. And we lose those cells. So that light sensing ability is gone. So what folks have done is that using gene therapy, there are other components that they're now putting into other cells in the retina that are still there. Cells, things that we call ganglion cells or bipolar cells. Their main job is not light sensitivity necessarily, but by way of using gene therapy with other components, they make those cells more light sensitive. And because it's not treating the underlying genetic problem, this is why it's gene independent. It can go for anybody that has the idea or has the retina right now that doesn't have those photoreceptor light sensing cells, and now we're going to try and convert other cells into light sensing to see if we can get more function back. And basically, that's what this is all about. And like I said, there are a number of companies going after this. It's in clinical trials right now, and we'll have to wait and see how this proceeds. The other thing is something called gene modifiers. And in genetics, it's a very complex space, but there's certain genes that don't necessarily correct the underlying genetic issue, but it modifies the effect of that issue that maybe we can restore some vision. And one of those things is a gene called NR2E3, and there are groups now going after that, that by doing gene therapy with that, regardless of what your gene is, that's causing your inherited retinal disease, it may be able to slow the progression of that. And so I'll take this opportunity to say that one of the most important things you can do for your health and your journey with inherited retinal disease or eye disease, well, especially with inherited retinal disease, because most of those are what we call monogenic or single caused by one gene, is to get genetic testing. And that would help not only yourself, but will help your physicians. Now, those of you that live with more like age-related macular degeneration, that is not a single gene issue. That's multiple different genes can affect, and there's environmental factors, UV light, smoking, diet, things like that. So that's a little different. But for those that have inherited retinal disease, getting a genetic test is really very key. All right, let's go to neuroprotection. Neuroprotection has been around a while, and there are a number of different factors that have been looked at. One is ciliary neurotrophic factor, pigment epithelial-derived factor, brain-derived neurotrophic factor, and then something called Rod-derived cone viability factor, and that's what we're working on. The other three have been tried, and unfortunately, they worked great, so one of them, CNTF especially, worked great in animal models, but it didn't translate the same effect when it came to human trials. And that's one of the things, just remember, mice are not small humans, okay? So just because it works in animals or models, it doesn't necessarily mean that it will work in a human. Matter of fact, I think one statistic said that less than 30% translates from a mouse to a human, okay? And but that is the model we use because they're much easier to manipulate, and to do genetic testing on and things like that. And that's a whole nother discussion about preclinical testing. But let me talk to you a little bit about rod-derived cone viability factor. And if I'm running behind or running on time, just let me know and you can yank me off stage here. But rod-derived cone viability factor was something that our founder, Professor José Sahel, he was at the Institute of Vision in Paris. He asked this basic question. In retinitis pigmentosa or rod cone dystrophies, we know that the genetic problem causes the cells in the periphery known as the rod photoreceptors. So let me just back up here a little bit. There are two light-sensing cells, there are two main light-sensing cells in the back of the eye. They're known as photoreceptors. There are rod photoreceptors and cone photoreceptors. The rods are in the periphery, and they're mainly for nighttime vision and your peripheral nighttime vision. In the center is your cone vision. This is what we use for reading, your daytime vision. This is your best acuity. And in rod cone dystrophies or RP, that tends to go at the end. But the question was, why do those central cells die? Because the genetic problem doesn't affect those cells. It really just affects the peripheral cells. And after 20 years of research, they were able to identify a compound that they call rod-derived cone viability factor, and basically saying that the rod cells secrete a neurotrophic factor that keeps the cones alive in all of us. It's a natural process. So the idea is, can we now take that and put it back into patients with RP who don't have those peripheral rod cells, that don't have that support of the central vision, and help maintain people with RP or rod cone dystrophies to have that central vision last maybe decades longer, or at least maybe until the end of their life. So that's what we're working on. And retinitis pigmentosa, this schematic has 5 different columns, and the first column shows a full retina, has all the rods and cones you need, and then the second one is when the rods start to die, and then the third one is when almost all those rods are gone, and now the cones are starting to get degraded. And underneath that, we show a picture of Renoir, because I work for a French company. So SparingVision is based in Paris, France. And yes, I have the unfortunate task of going to Paris about 8 times a year. So I know you're very sympathetic on that. And we have an office in Philadelphia. But what happens in this photograph of this beautiful picture by Renoir is then when rods and cones are fine, you see the whole picture. But as you progress, you start just seeing the central part of her face. And then after that, even her face starts to get blurry and the colors start to go. This is what happens in retinitis pigmentosa. And so what we'd like to do is try and use gene-independent ways where we can actually prolong that central vision so you have the ability to still be able to read and ambulate and things of that nature. So how does this work? So rod-derived cone viability factor is something that is secreted by those peripheral cells known as rods. And it goes through a whole bunch of different steps, and I'm more than happy to discuss this with you after my talk because it gets a little in-depth, but there's a whole lot of metabolic pathways that happen. It increases the amount of sugar that comes in. That sugar is used to form carbohydrates, and this maintains the function of those inner cone cells. The cone cells are the ones, again, that we use for color, daytime vision, and for your best acuity. And so it's basically replacing what is lost from the loss of those peripheral rod cells. So where are we in this? So there's a schematic on the screen right now, and basically what it's depicting is the clinical trial. And the clinical trial is what we call a phase 1/2, where the principal component of this is to prove that our product is safe. Of course, everyone wants to know that it works as well, but our primary goal is to make sure it's safe. And this is divided into what we call a dose escalation. So we use 3 different doses in a basically ascending pattern to see what dose would be the best for our patients and the safest. Once we have done that, and right now we have finished that. Matter of fact, we just dosed our last patient last week, so we're all very happy about that. We go into an expansion phase where we'll use the best 2 doses and then what we call a control group, or a group of patients who will not receive anything. And we'll follow them and compare them for 1 year, and we'll see if there are any significant changes between the different groups. So with that, I just wanted to mention, where are we in that when it comes to safety? So right now we have the data for the first 2 cohorts, and the safety has been very favorable. We haven't had what we call a serious adverse event. We haven't had something called a dose-limiting toxicity, which means that there is some really adverse event that causes us to really stop the trial in general. And we haven't had any other real issues with that. And that's been both in our low and medium dose. The things that we do see is that things associated with the surgery. And because it is a surgery, so a lot of times gene therapy is done through what we call a subretinal injection. You're brought into the operating room, you're put under anesthesia, and basically, and I hope nobody's real squeamish here, but we take a needle and we put that virus underneath your retina. And luckily, this is also the way Luxturna is done. It's a very complex procedure, but in the hands of your well-trained vitreoretinal surgeons, it's commonplace. So we're not worried about the procedure, but anytime you penetrate the eye, there are possible things that could happen. Now, for us, we've been fortunate that they're minor things, such as the person gets a red eye, that might be a little sore, the idea that there's just a mild amount of inflammation that goes away on its own. The pressure in the eye goes up a little high and then comes down. One of the things we do is we take the jelly-like substance that's in the back of your eye, that's called a vitrectomy, and that can also accelerate cataract production. And so those are some of the things that we have seen. And I have to be honest, I have to step down for a minute here to get a look at what's in there, but that's kind of what is on that. So right now, we are really looking at the safety of this. We're very happy that our last cohort has been done, and we will be following them, and hopefully later this year, we'll be able to start the expansion phase. So we're very happy about that. On the panel right now, there are 4 different graphs, and each one of these graphs represents a measure that we look at, and that is inflammation. Gene therapy in the eye, this is probably one of the biggest issues or non-issues because we've been able to remedy it. These patients all get steroids prior to treatment. We've been able to dampen the inflammatory response. And in these 4 graphs, it simply shows that you see a little spike right after surgery, but then it goes right back down to baseline. So that profile has been relatively good. Now, there's a second program we're also working on. This is using something called GERK. GERK is G protein inwardly rectifying potassium channel gene. And what does this do? I wish I knew. No, I'm just kidding. The reality is that as you start to degenerate and your central cells start to die, so this is very end stage, very severe disease, is that those central cells, they lose a functional part of it. It's called the outer segment. I always say it's like the hand waving in the wind. It's a very active area. This is where a lot of the components of converting light that comes into your eye, it goes through a whole bunch of processes and it's converted into an electrical response. And that response is sent to the brain, and the brain says, you see, I'm sorry to tell you, but your eye does not see a thing. It's your brain that sees. Your eye is a complex relay system of neurons, and it has to be intact to send that message to the brain. So here, that message is not getting sent. That, what we call phototransduction, is not being sent. And so can we, with that, can we restore phototransduction in these cells that have lost that light sensing apparatus, that outer segment, but still have the rest of the cell intact? We call that a dormant cone cell. So the other program was working with folks who still have the cone cells, they don't have those peripheral rod cells. Let's try and maintain those cones. In this, the group we're going to start with are folks that have absolutely no vision. They're what we call no light perception. And the idea is, can we restore this idea of phototransduction in terms of getting that light coming in and converting it to an electrical response so the brain can start to say, oh, I get a signal now, and we get some visual sensation. Obviously, if that works in that population, the goal is to go to those who have a few more cells and things of that nature. Now, you're probably asking me, patients who have no light perception, do they really have any cells left? Well, the reality is that in studies done both in Basel, Switzerland, and in Paris, France, anywhere between 30 to 50% of patients who have no vision whatsoever still maintain those cone cells that have the cell bodies still intact. They don't have the light sensing aspect, but they have the central body or the central cone cell. And the idea is through gene therapy, we will put this into those cone cells, and maybe we can get some light sensitivity or more back. And because there are other components that we call opsins, this is really what is good for color vision. Maybe we'll be able to get some color vision back as well. Now, I do want to mention, somebody asked about research and where are we? Well, we're on clinical trials, which means we don't know if this is going to work yet. And the way clinical trials run is that you have multiple phases. You have phase 1, phase 2, and phase 3. And many times in rarer diseases, we combine phase 1 and 2, or we don't do a phase 2 at all. We do a phase 1 and then go to a phase 3. And so what is a phase 1? Phase 1, as I mentioned, is really about safety. Phase 2 is also safety, but then you start thinking about efficacy. Is this really working? And then phase 3 is what we call our pivotal or registrational trial. And that one, you have to pick what test am I going to use to prove that our drug works. So that you got to pick before you even start. And then all that data is used to go to your regulatory bodies, whether it's the EMA, Health Canada, or the FDA, and they'll use that data to say, we approve this product and it's safe for patients. You can go ahead and get it to the patient. Let me just tell you, that's not the end of the story, though, because then we have to work on the payers, because it's hard to put out that money on your own, so we work with payers as well to get that product to you. And I think that was one of the things in Canada. The product was approved in 2020, but to get somebody to actually pay for that, it took a few more years. And so the journey isn't over until the patient who needs it gets it. That's when the journey is over. That's when I feel like my job is done. So that's a big tangent, and you'll know this morning if you were in that session, for those you know I'm a tangent person, and I can go on a tangent forever. How much time do I still have, by the way? So, 7 minutes. All right, very good. All right, so this is the next program. We hope that we'll be in the clinic in this in 2025, and we think that this is something that is restorative. So in other words, you're going to go from nothing to something, whereas the program before, the RDCVF, this is about slowing down. And that's a much harder bar for us to prove. But in the end, it could be worth it. So for the GERK product, it's just an injection into the main part of the eye, not a subretinal. So anybody who's getting those AMD shots for the anti-VEGF, this is going to be the same way. It's going to be outpatient. You won't be in the operating room. It's going to be a much simpler administration. So with that, what are the take-home messages? So I'm going to come down here. A gene-independent approach, it gives us the ability to treat a much broader population. And in that, as I said before, there are some targets that are so rare, nobody can develop it, because in industry, they always look at how many patients we're going to treat. I have a very difficult time with that because obviously I worked on Luxturna, and that's a very rare indication and is changing lives. But that is something that we can now do if we do a gene independent. The other one is that we can also, and this is more practical, it'd be a lower price point because it's treating many more people and it's not restorative. And in a time where, I hate to even talk about it, but the reality is to get these products to the market and everything, to go through the whole, you need resources and we need investors, and that's also attractive to them. This is not a competition with single gene therapies like Luxturna, okay? It gives us another tool to help you make a good decision. So if you came to me with an RP65 variant, I would say take Luxturna. Don't do our gene-independent program. Do the thing that is targeted for your gene. It gives us, in the end, another shot on goal. Wouldn't it be great to go to your doctor's office and have an option of what you can choose from? And that's my whole point, is let's get more shots on goal, and you and your physician can make the good choice for what is right for you. So with that, I do appreciate your time and happy to take any questions.
I'm going to come up here. Thank you so much, Dan. For anybody who doesn't know, I'm Dr. Larissa Moniz. I'm the Director of Research and Mission Programs at Fighting Blindness Canada. And now we're opening up the conversation to the audience. Do you want to actually
Sure.
over there, sit down? So I think we have Jackie and Jamie, my two colleagues, have microphones. I can see hands up already, so I'll turn it over to Jackie for the first question.
My question is, what is the long-term effects of the therapies? Does it cause long-term effects that you're not aware of, side effects?
So the reality is we don't really know exactly what that entails. If we take for Luxturna, for instance, because right now obviously it's the only gene therapy for the eye, and right now we have about 10 years of data. There are things that can pop up that we don't see in clinical trials necessarily. In Luxturna, there is something called choroidal retinal atrophy that affects about 10 to 15% of patients. And what we have found that right now, it hasn't really affected a lot of the functional improvements, but it's something that we're going to look for long term. Now, for the safety of it, the inflammatory response we think is relatively well controlled because right after surgery and things of that nature, you're on steroids, and most patients, even after treatment for many years, there doesn't seem to be any detrimental effect. So right now the data says that for the most part these are relatively safe. And remember, there is no drug out there that is totally safe. And that there will always be some, unfortunately, some folks who will have some adverse reactions. But right now, for the most part, it is what we say is a favorable safety profile. But it's one of those things that you have to really talk to your physician about and think, is it for you? For many of the folks that have taken Luxturna, the alternative is they know they are going to get severely more and more decreased in their visual function without anything. And with Luxturna, they've actually been able to restore some of their visual function, and that has been maintained for some time. There is no perfect drug. It's the first gene therapy out there. It's not perfect, but it definitely has changed lives.
I actually had just a follow-up question. As you were talking before and also right now about inflammation, I was wondering if you could just explain why inflammation is so bad and why that's a key thing you're looking at.
Yeah, so remember, the eye is a closed organ, and anytime you penetrate that, whether you're doing cataract surgery, vitrectomy, or any other procedure, you're going to get some inflammation. It's the body's way of reacting to this insult. The reality is that you need to try and control that, and usually we use steroids or non-steroidal medications both during surgery, after surgery, and most of that is kept to a minimum. But if you do have inflammation, it can have a detrimental effect that it can affect the structure and the function of the eye, and if you have a significant amount of inflammation, it could lead to other complications that can definitely be vision threatening. So right now, AAV is what we call adeno-associated virus. This is the carrier, this is the vector that carries that gene to the cell. And for the most part, AAV is relatively safe. We haven't had a lot of issues with inflammation, but as I said, there will be a portion of folks who may not react well to it, and those are the ones that we keep an eye on to really try and make sure that we have the right medications on them.
Thank you.
Thank you. My sister is one of those people who have no vision left, and she did try and get into the optogenetics trial. So my question is, because she didn't get in because she has a syndrome, who sets the criteria for the trial, and what are the chances of getting some of those changed? Like, would it change if they didn't have enough people, or what? Yeah, I just wanted to understand the process.
Yeah, clinical trials, basically, we as the industry is really the ones who set a lot of those parameters. For myself, I always consult some of my colleagues in the academic space because they're the ones that are seeing these patients on a daily basis, so we try and get their input as well. Remember, in a clinical trial, we're trying to keep the patient population as homogeneous as we can because if you have a lot of variation, then the signal you get after the trial can be very misleading, or it can be confusing, or there's not a really consistent message. So you have to fit a certain criteria in the clinical trial itself. Now, if you're fortunate and that gets approved later, it becomes much broader that you can achieve. But that's why clinical trials tend to set rigid standards, because we're trying to select and minimize what we call the heterogeneity of the population. So that's probably what that is all about. Now, for us, we will probably be a little more broad when it comes to the NLP population, but even with that program, there are certain things that we're going to have to exclude because there may be certain interactions that may occur that we want to safeguard against. So remember, phase 1 trials really about safety, so we really want to make sure that we're doing everything we can to ensure the safety of the patient.
So sort of following up on that question, you mentioned once a treatment is approved, it might be able to go broader, the eligibility criteria. Is it hard, though, once you've done trials in a certain population for the regulators, whether that's Health Canada or the FDA, to say, oh, actually now you can treat people who didn't fit that criteria, or are they pretty understanding?
It really depends, again, on what you're talking about. For gene-specific trials and things like that, you're not going to get indication other than those patients that have that specific gene. So what happened with Luxturna is we did our trial. We tried to enroll 3-year-olds and older. 3-year-olds really liked the maze as a playground and not as a test. So we didn't get many 3-year-olds, but 4-year-olds we were able to do. FDA actually said, okay, that's fine. We're going to actually allow you to do 12 months and older. So it's a broader population that way, but it's always going to be an RP65 disease. So in gene-agnostic, it's really the idea of how many different genes do we have to test and treat before the regulatory bodies will say, okay, you have enough data here, you can do it with anybody that has rod-cone dystrophies or things of that nature. You know, for us, we use 3 different genes right now because those are the ones that we had animal models for, and so we had to prove that it was safe and that it worked in animal models, and that got us into the human trials. But with only 3 genes, you know, they may not be like, okay, go ahead and treat everybody that has RP. So we'll have to do more trials, most likely with other genes.
Thank you. I think there was a question from the live stream. Do you want to go to that next, or should we go elsewhere, Morgan?
Nope, I'm coming back. We have a few questions online. So let's go with this one. Is SPVN20 limited to RP patients, or can it be used where there are dormant cones?
So SEP290, is that what we're talking about?
No, I think SPV.
I think they were referring to gene therapy, SPVN. Yeah,
SPVN 20. These are both gene independent. So it really doesn't matter what your underlying genetic cause is. We would still like to know what your genetic cause is, but it's not dependent on your genetic cause.
It's more important that you have cones available.
Correct. So we would do something called an optical coherence tomography, which is a way we take a picture of the layers of the retina, and we can see that you still have some residual cones left.
We have a question. Jackie, back here.
Yep, just coming over here, sir, to your right. Here you go, I saw your picture.
Thank you. Yes, thank you. Just wondering, I was initially diagnosed with the retinitis pigmentosa many moons ago, and eventually I did a blood test and they changed it to choroideremia. Now I'm hearing all of the RP65 thing, and I've heard about the trials, which I applied for and was rejected because I was too old, I think it was, their term. Anyway, I'm just wondering, is choroideremia an RP65 thing? Have I got the same thing, or is this a different thing altogether?
Yeah, it's a different disease. RP65 is kind of overlaps. It's what we call a labor congenital amaurosis, or early childhood onset, and then a very, very few folks I think have been documented as actually having RP with RP65. Choroideremia is with the CHM gene, and it is a rod-cone dystrophy, so it follows the same pattern as RP, but the retinas do look slightly different in the way they present, different gene mechanism. So it is in a different group than retinitis pigmentosa. And it really depends on who you talk to. a lot of people lump everything in the back of the eye that looks genetic as RP. I call those the lumpers. And then there's the splitters like me, where there's RP, there's choroideremia, there's LCA, there's, so everyone is a little different on that. But choroideremia is different than RP.
Okay, we have 2 more questions at the back.
Yes, I seem to understand that gene-agnostic therapies are kind of generalist in their approach. Is this an accurate assessment? And if so, do you work with companies that are more specialized in the type of diseases that they solve?
So the approach is, if I'm getting the question, the procedure in which we do the gene therapy is still similar to those that are, even for like Luxturna. It's still a subretinal injection. We still do genetic testing. We do use a lot of the same endpoints. For us, we're trying to maintain central vision, which are the cones. So it's a little different than Luxturna. Luxturna is more about the rods and peripheral vision and light perception and things like, or getting more light sensitivity. So our tests, some overlap and some are more specific for us and some for LCA. So, the community is relatively small in the IRD space. I've been in this space since 1994 when I did my fellowship at the NIH. And so we tend to talk frequently, but whether it's gene agnostic or gene specific, the approaches are relatively similar. And we still want to engage in patient organizations. We still go to the same conferences. So there's really not a lot of difference in the approaches. It's just a little harder to prove gene agnostic versus single gene.
Okay, I'm going to go over to Jamie first and then back to Jackie.
Hi there. Thank you so much for your presentation. I understand that to be enrolled in clinical trials, you typically need to have a genetic confirmation, even if it's a gene-agnostic therapy, which makes sense when you're talking about homogeneity of patients. When these become available to patients, do you think any of these gene-agnostic therapies may still need a genetic confirmation to be eligible to receive them in the clinic?
Yeah, I think that's a great question, because obviously for gene specific, you definitely need a good genetic test to make sure you would qualify. I think I'm always going to be a little conservative. I might be in industry, but I still consider myself somewhat of an academic. And so I want to know what your gene is before I put you in a clinical trial, even if we're gene agnostic. Once it's approved, I think that if it's for RP, then your payers may want to make sure that it's an RP gene. Now, eventually, we like to go through all rod cone dystrophies, which would include choroideremia and other entities like that. So I'm never going to come up here and say you shouldn't get genetic testing. We have worked very hard to make genetic testing part of the landscape. So I would always recommend genetic testing. Now, even with genetic testing, only about 60 to 7% actually get a diagnosis from genetic testing. So there still is that, but so kind of a mixed answer there. It's going to be kind of what the payers want. I think for the scientific and medical communities, it might be different. I always think it's good to have a genetic diagnosis.
Thank you. Is there any research done on vision loss caused by optic neuropathies?
Yes. Optic neuropathy is one of the more advanced ones. It's something called Leber hereditary optic neuropathy. There's a company that has done some phase 3 trials. I think they're going to have to do a few more phase 3 trials. Their efficacy profile looks favorable, but it's really more about how they did a clinical trial and things like that that didn't quite cut the mustard. So I think there's more work to be done there. I know there's also a group for dominant optic atrophy that's also being looked at as well as some of these mitochondrial diseases and things. So yes, there's a lot of work on that. LHON, I think, is probably the most advanced in its program.
I have come to this kind of conference many times over the years, and I will tell you, your presentation is a more complex one. I
hope that's good. So.
Well, it just seems to me that the whole field of vision is getting more and more and more specific because the research is getting down to the finer detail. But it isn't really encouraging research, in my opinion, what I have heard today, in that there are so many genes, you're dealing with one, and even with dealing with only one, it is still so complex, it's barely understandable to the general person with a vision disease. I'd like to know, with the work you're doing, is it with a gene that is relatively common, a vision problem gene that is relatively common, or is it a really sort of rare one? Because I don't remember, I think you talked about dozens and dozens of genes that can lead to a specific problem and a specific treatment. Am I on the path, or am I a bit off track?
So I think the one thing I did talk about was Luxturna, which is a single gene product, which is for a small group of folks with inherited retinal disease. What we're working on right now, though, is a gene-agnostic approach, which means it could treat, if successful, anyone who has RP. So it's not for a single gene product. And so it would be very much a vast population that we could treat. We treat, if it works, and I put a big caveat, if it works, then anybody that retinitis pigmentosa could be treated by it. And that's why we're looking at this as a gene-agnostic way instead of a gene-specific way, to your point, that it's one gene, and it's great for those people who have that one gene, but what about the rest? And so through gene-agnostic or gene-independent therapies, we're not using a specific gene in terms of, we're not changing the underlying genetic issue. We're using something that is what we call a neurotrophic factor, which means it protects the neurons in the retina from degenerating more. And we do use a gene, but it's not a gene that causes an inherited retinal disease. It causes basically, it's a gene that helps protect your cone cells from dying. And that's what the gene-independent approach is. So exactly to your point, is that we are trying to use one approach to cover 80 or 90 different RP genes.
Penicillin, that's like your penicillin for eyes.
Correct, yeah. Yeah, hopefully it won't go the way of penicillin with all the intolerance that it's having right now, but you know, if we can just do that, because then, like I said, it can treat a lot of the genes that are so rare, they'll probably never get developed to a point of treatment, so.
Okay, this is going to be the last question for now, but Dr. Chung will be around, so if you don't have your question asked, I'm sure Dr. Chung will be happy to answer it in the break.
Great.
Thanks
so
for
much.
coming today. Is a defect in the retinal pigment epithelium still thought to be important in macular degeneration, and what research or clinical avenues are open in that regard?
Yeah, AMD is one of those complex diseases, what we call polygenic, environmental. There's some genes that will predispose to you. In other words, if you have a defect in that gene, it increases the likelihood of you having AMD, but it doesn't guarantee it necessarily. And there are multiple genes like that. So RPE cells are the cells that are right next to the photoreceptors, and to me, they're used, I think of them as somewhat like the trash cans of the eye. They're responsible for all the debris that comes off these outer segments, because the outer segments are constantly changing and renewing. And the RPE cells are important for that, and part of that whole light to electricity conversion happens with the RPE and the photoreceptor. AMD right now, especially in dry AMD, they are saying that those photoreceptors are dying very early on, and that maybe it's not just RPE, that it's also a photoreceptor disease. And with that, we're actually looking at our product, the RDCVF, because we're trying to save cone cells, cone photoreceptors, that we probably will be going into AMD trials as well with that component. So it's kind of a mix right now. I would say that there's a lot of literature out there that it's a photoreceptor disease as well, but right now it's really known as an RPE cell disease, but I do think there's a lot of discussion and change on that.
Okay, I just wanted to thank Dan so much for coming. This was a wonderful session.